Okay, yeah. Hello, everybody. Again, I'm Bogdan Parakhonskiy, and now I continue with my second, or with our second presentation, because first part we talk mostly how to how to design the biocarrier and to mix the particle cells and so on. But here we try to talk about one of the technique which are very interesting to functionalized kind of particles, bacteria, cells, like the microfluidics. Yeah, and we'll talk here about the materials and fluidic design to study the artificial functionalization of the microorganisms. Okay, but it doesn't work. Oh, yeah. Okay, in the case in this, it will be more kind of technical presentation because it's about methods to give you kind of introduction about the microfluidics, what it is about and how to possible and how mostly in other groups to apply them to research bacteria and about the mechanistometry and what we have in GHENT about these techniques. One part of them I will talk, give a talk about mechanistometry and yeah, our part, Professor Skirtach will talk later. Okay, then kind of introduction. The microfluidics, really, it's kind of interesting area because it's combination of the science and technology. And in this area, really, we are, I mean, all microscopy and all spectroscopy techniques, really, it's combination of the science and technology. But here it is, we have feeling about it's much more stronger because we can manipulate with the fluids, and behind this there is kind of a lot of equations of the theoretical mechanics and mechanics of liquids, but also a lot of design, cheap and engineering to provide the kind of real application of them. And really from real application, if we are not kind of theoretical liquid physics, it's mostly engineering part, how to design real components and why we need to design what we'd like to end in the end. Because the components and the engineering part, it's much more important here than in rest. Yeah. What's the idea of the microfluidics? There are kind of different layout and yeah. Oh, I think here already this works that okay, it's manipulation with the fluid liquids, with the fluids in the micro level. But there are really 4 general kind of branches of them. It's first of them, it's continuous microfluidics when we mix just 2 liquids in special conditions and get as a result the kind of soft liquids. The paper microfluidics, for example, when we have special reaction in paper and the liquid penetrates through the cellulose pattern, for example, to react. Also, the digital microfluidics, which we can manipulate with the liquids in a local level by the chips by provide the kind of current and in case we mix, split, and so on. But more important for our talk, it is a droplet microfluidics. Since we're interested to apply them for colloids, for biocolloids, for bacteria, microorganisms, or the particles, all of them, it is a kind of colloidal system. Yeah, in the case, it will be a kind of droplet microfluidics because, yeah, or we need functionalized, or we need to have micro reactor about it, or we need split or separate them. In the case, yeah, in my presentation, I will talk about the droplet of microfluidics because, yeah, the idea behind it, it's kind of enable high throughput serial generation of the aqueous droplets in a carrier or oil flow. Because really the whole idea about the kind of fluids and generate droplets, it's already exists before microfluidics. You can put the drops from the pipette one solution to another solution and so on. But in upscalable and really repeatable nano level, it's possible to just achieve with a microfluidic device. In the case, in a lot of labs, just like in we dreams to do our processing, our reactions in a controlled small volume liquid, which will be repeatable and completely controllable. Yeah. Really, the microfluidic has many applications, but a lot of them it's already applied. But as I said, the droplet microfluidics is more interesting because just we can have the rapid cell encapsulation, a rapid functionalization of the bacteria, and it's more precise. But for this one, we need kind of droplet generator, sense droplet. Droplet generator, usually it is much more quite easy substance. You don't need to have kind of special chips or pumps. You can just combine few flows. One of them will be oil, another one will be water. In the case you can provide the oil bubbles. In the case if you premix well with kind of another substance, you can cover them, you can increase the frequency, increase the speed of your pump, and influence to the size, shapes, and so on. Really, if we go to the more technological part, as I said, the chips here, it is very important and more important part. In the case, depends on your design, you can provide different kind of functionality. For example, the typical T-junction can provide the separate drops. If you have flow focusing, you can encapsulate something. If you have co-flowing, also you can have one drop inside another one. If you have chip which has special geometry, you can cut your big drop to the small drops. And all of them explained between the fluid of the not mixture liquids like the oil, for example, and water. In the case you have two liquids, one inside another one, which has a different behavior under the pumps. What is more important here and how they usually it's characterized at the in typical 10 junction, when you have two solutions and you have the aqueous stream and you have oil which cuts them, you have just two parameters. It's the concentration of the oil, or not concentration, just speed of the oil, it's speed of the aqueous stream. In the case, depends on this, you can generate different sizes and different geometry. But really, it depends on the chip configuration. You can operate with different, operate in different ways with your droplet. You can provide kind of fusion of the materials in a kind of special small chamber in your chip, or you can split if you put the two channels in separation, or merge to and generate new bits, for example. Now I will step by step talk about how they realize in practical. For example, for the drop merging, you can put in your chip just small grid. In the case a drop will be stuck on it and merge each other, for example. Or you can apply kind of pressure and put one drop inside another drop. Also, depends on the your particles, your drag inside the drops. It depends how they are heavy or not. It could be circulate or also influence to the drop geometry and constraint in different way in drops. In the case, for example, if we had the kind of magnetic particles in it and segregate them with add to the such alginate or kind of bit the magnetic nanoparticles, we can gather and manipulate with our bits by the magnetic force. In the case, we can add additional functionalization of them, because it should influence to the flow as well to the final object geometry. Also more important, what also should we do with this droplet which we generate by the microfluidics? We can do kind of transport manipulation, and I mean the take our drop with the tweezers and move somewhere or something like this if our chip allow to do it. But very interesting, very important in the kind of sample separation. And really kind of sample sorting inside the chip, or design the chip for sample sorting. It's very important task because it can help to sort you what it's already functionalized, what is not functionalized, what in your system can provide kind of property or can provide kind of special adhesion and so on. And usually there are two types of sorting. By one of them it's by chemical composition, other one by size. Yeah. Really, by the kind of size, it's much more easy because, okay, or you put the filters, or you can put the separators, or it's more heavy, you can provide kind of gravity separation, or you can put kind of pillar array when they not pass. Yeah. But really, if it is by chemistry, it's more precise and more interesting. For example, I'd like to show kind of one example how it's separated by the carrier which can adsorb or not adsorb the surfactants. For example, we have a droplet stream with our carriers, and we have carrier surfactant. In the case, if it's adsorbed, they go to the special way on the chip with a very surfactant flow. But really, we can adsorb in kind of way our enzymes. In the case, our enzymes, protein could be inside this flow, and with a schematic like this, it could be water with protein, and we can oil drops with a protein. And if it's adsorbed well, of course, depends on the speed, we can also check it. For example, if the droplet without protein, it could be flow by the surfactant and to go with surfactant stream. If it's with protein, surfactant will be not adsorbed because it's already surface already saturated with the proteins. In the case, based on this idea, in kind of already many years ago, it's realized, and I show them as example, it's possible to separate the put amount of the different containers or carriers in the stream and check which of them will be adsorbed better the protein, and sort which are adsorbed or not adsorbed by based on the chip reaction. Also, really, what is more important and what we try to kind of realize partly also in our lab, it is a kind of imaging techniques of such microfluidics which require kind of very high speed camera and kind of big kind of important image analysis, because we can record how we construct kind of new objects, what size, shape, depends on kind of conditions, flow, and so on. And after this, do the image analysis on them, and to understand the influence of the final objects immediately after the synthesis. Because now, if you synthesize normally, we need to do after this, do the electronic microscopy, optical microscopy, and it's do separately. But here it is by analyzing the streams, we eliminate completely the storage condition, the sample preparation. We can analyze immediately what happened after the particle synthesis. Yeah, and more common way how really we can find the microfluidics approach in this workflow cytometer idea. In the case, if you have kind of different cells, bacteria, viruses, and we separate them encapsulated by oil, and after this we can check by optics and apply the and sort them using the electrical force. And in this case, yeah, which encapsulate can go in one direction, which encapsulate don't go to another direction. In the case, more or less the same idea in flow cytometer, it's already realized. Yeah. Okay. In the case, really, how we can apply really or few ideas how it's possible to apply such microfluidics approach to the bacteria. One of them, which we found in the literature, it uses such microparticles with the microreactors. In the case, we can put the agarose and bacteria mix with agarose and create the droplet of agarose and check how the bacteria grows in exactly one droplet. The positive stuff here is that it will be small volume, and we can add more components on them and influence really for a single bacteria how this components influence exactly to this one and not to the total mixture. And I think this one very important, interesting application also for us to try to generate it and add more functions. Second one, it is the kind of bacteria functionalization. Also, it's already exists in the literature, but it's possible to pass the bacteria through the microfluidics, put the kind of, for example, magnetic nanoparticles, and if it's magnetic nanoparticles adsorbed to the bacteria during this generation, we can modify them in very controllable way in very kind of thin layers. Yeah, and kind of one of last application, which also interesting to produce, but as you saw here, we need much more fancy, cheap, and design well. It is a kind of microfluidics for surface enhanced Raman spectroscopy. The idea is here that we have kind of bacteria, we have channel with bacteria, channel with oil, and channel with a kind of silver colloids. In the case we really in a chip, we absorb the silver colloids on the bacteria, and immediately saw the detected by the Raman from the single bacteria to understand them what reaction on surface or gather the data. Really, from this paper, it's okay, already 10 years ago, they do it well, and they show that the selectivity and so on much more better than the simple Raman, and that just simple cells as well. Because really here it is, you can detect something from the exact single bacteria, and amount of the silver which absorb on bacteria, it's very controllable, and the signal acquisition, it's also very controllable. Yeah.
Good afternoon. My name is Skirtach. We'll continue this talk speaking about mechanocytometry and highlighting capabilities that we have in our own laboratory in regard with microfluidics. So mechanocytometry is a relatively new area. We are talking about several years ago. Yes. Several years ago, mechanocytometry was reported as a mechanobiology analog of flow cytometry data. So in this slide, you can see data taken from a recent publication. This is not our work, but
sorry to interrupt. Sorry to interrupt, but you lost the screen sharing.
Sorry, where is it? Yes, yes, I was alluding to a very simple schematic here. Here in the upper left corner, we can see a small constriction. And in this constriction, under flow, cells go one by one. And as we can see here already, there is a small deformation in the cell shape. This is generated by the pressure difference of an aqueous solution in which cells are located. So if cells translocate through this constriction, what is being monitored is the so-called circularity. So normally suspended cells are circular, but if pressure is applied, and it's applied non-uniformly, then part of cells will deform more than other parts of cells. So there is a deviation from cells circularity, and here some brief outline of this cell circularity is shown as a function of displacement. So cells are more circular in the free aqueous solution, but in this smallest constriction, they experience or undergo largest circularity change. And we can see here also some data when the cell is being pulled through this small constriction with the different colors. We can see stages of translocation. So indeed, the cell is mostly deformed when it is in the constriction. Some modeling was done, again, this is not our work, but some modeling was done on looking how a cell is deformed. So we can see here, this is a flow. And in this flow, you can see now a cell resembling a bullet. So it's sharper at the beginning, and it has a little bit of a flatter backside. So this is a schematic that we see here. Circular cells, but once they are dragged in this smaller area constriction, then they deform, and the circularity deviation will provide information about mechanical properties of cells. And cells do deform because their lipid membrane is largely mobile and very flexible. Implementation of these devices typically taken implementation of mechanocytometry was taken place in a microfluidic setup. So this is a side view. This is a glass. We see here this material that makes microfluidic devices. This is PDMS, low temperature transparent material. And then view from the top, we can see here that there are some areas where cells are not pressed on, and there are this areas in the middle, constrictions, and we can see microscopy. Everything is easy in regard with microscopy. This is the transmission microscopy images. When we look here at cell deformation and deviation from circularity. One goal of this mechanocytometry study is to study mechanics, softness and hardness of cells, or mechanobiology, but at a very high throughput. In the high throughput, another technique that pops in mind, and this is flow cytometry, where side scattering and forward scattering detectors tell us tell information about the state of cells. Here in mechanocytometry, typically a deformation is a deviation from circularity is spotted versus cell size, and depending on the cell size, their stiffness changes. So there will be a dependency here. And we can see in the next slide, we can see that different cell types were put through this microfluidic constriction area, and they exhibited different deformations as a function of their cell size. From the same publication, basically an analogy was made between mechanocytometry and flow cytometry, and you can clearly see here each pixel represents one cell. Cells are different, so you see that there are some a large number of cells covering the whole area, but we can also see some clusters, and this is most important because based on these clusters, different types or different phenotypes are determined and classified, again based on the deformation versus sizes of cells. In the next section, we will highlight, or we will show what's available at our group. And you will not see that in the publications. You will not find this in the publications yet. But we are preparing some diversified range of applications. Some of them pertain to making particles, but some of them also, in some of them, investigate deformation of cells. So in microfluidics, we typically need some imaging system, and this is a key. So very often a microfluidic setup is being placed on a inverted microscope, or actually upright microscope would also be suitable. So then it's very essential that the fast camera is connected, because we have many images are generated per second. And then a chip, this is a key part of microfluidics. So a chip needs to be designed depending on the number of mixing liquids or number of different components that are generated in microfluidics. And then we need also to supply of other active materials, or maybe cells or bacteria. So this typically done through plastic tubing connected to this controllable syringe pumps. In this case, we refer to this setup as a manual setup. Manual, it means that we manually set up the syringe pumps. What you set up here is one setups here a pressure on this syringe, and by this we can control the flow in the channel that we generate. But we also have an automatic setup. So in an automatic setup, then we speak not about syringe and syringe pumps, but there are some controllable pumps, and there are a number of controllable pumps, and these controllable pumps are used to create a pressure difference. Again, here in the chip, different channels here are connected to different vials with water. And we typically take some sample from here, from green, and there is some admixing takes place in this chip. There are two arms that we can see that this admixing area, and then we generate finally solution that will be collected in this vial. So the difference between manual and automatic setup is in place of syringe pumps. We have here an automatic pressure unit. Here are some images of the chips. Again, chips are the key parts of any microfluidic setup. So we can see here two simple ones, two admixing channels. Here we can see outlets for pipings. Pressure is applied, water is put through, maybe cells or bacteria can be circulating here. And then active interaction area, here it's been colored in blue. This is a setup in action, so we can see here some vials and some collection reservoir. This is a microfluidic chip, and then pressure is controlled by the unit, and admixing takes place here. Yeah, so this is again we refer to this as an automatic setup because pressure is controlled here. These are some, let's say, preliminary data from our laboratory where microbeads were generated in the chip, and you can see here one by one on the this large scale. The chips one by one beads are generated, and then they can be used for either creating new particles or encapsulating bacteria, or for that matter, analysis of bacteria. And then it is very interesting now because we have just obtained recently some preliminary data, but the idea is to apply this setup for a broad range of either analytical calculation or fabrication of particles, beads, or analysis of cells and possibly bacteria as well. So with this, we would like to conclude that microfluidics is a largely engineering area that lies in between science and engineering. The chip is a key here. So depending on the chip, chip will enable you to analyze or generate different particles or beads. Various layouts are possible, but very commonly spread setup involves droplet generating microfluidics. Typically, chips are generated by PDMS material, that's low temperature melting gel that's typically mounted on a transparent glass, so everything is transparent here. So there is a large number of applications that's some of them are reported, some of them are in development. One interesting application is mechanocytometry. That's a recent application, but also very promising to investigate mechanical properties of cells at a high throughput. So we do have in the laboratory both manual and automated microfluidic setups, and we have already obtained preliminary data on the bead generation, and also we do have some preliminary data on mechanocytometry deformation of cells. And again, this is done at a high throughput. So again, interesting time because we are at the beginning of applying this technology. So this could be also done in the frames of our project. Thank you for attention.